Good morning, class. In the previous class we had done origin of life. Today we will be starting with evolution. Evolution is a term that was coined by Herbert Spencer, and evolution is a naturally occurring process. Basically, evolution is a slow, continuous, dynamic, naturally occurring, irreversible process of change. By this we mean that evolution takes it for a complex form of life to be formed from simpler form of life. It took millions of years. So we say it is a slow, and over this million of years slight changes kept on taking place. So we say it is a continuous and a dynamic process. And what has taken place today, what forms of life that have been formed from simpler forms cannot revert back to those original simpler forms. So we say it is an irreversible process of change. Now, these simple forms of life underwent some kind of variation. There was change in their chromosomes, in their genes. So we say there were chromosomal variations, there were genetic variations, and because of the ability of these organisms to adapt to their environment, that is what we say, those that were fit were able to survival of the fittest, given by Charles Darwin, and that's how these fittest organisms were selected naturally in nature. So we call it natural selection. Evolution results in the formation of a new species which are better than the older species. All organisms have evolved from a common early form of life, and there are various evidences of evolution. Evidences from comparative anatomy and morphology. We will be discussing these evidences. First, evidences from comparative anatomy and morphology. You know, anatomy means the internal structure of any organism, and morphology is the external structure. So the similarities among the different types of organisms can be traced back on the basis of structure of organs and functions of organs. So on this basis, there are four types of organs: homologous organs, analogous organs, vestigial organs, and atavistic organs. We have to do two main kinds of organs: homologous and analogous organs. Homologous organs are those which show structural similarity. That is, the structure we will see in different kinds of organisms. The structure of a particular organ is same, whereas the function performed by that organ differs. So such organs are called homologous organs. So we will first do the homologous organs in evidences of evolution. We have to do two examples from animals and two examples from plants. So first, homologous organs. Homologous organs are similar in structure. Now, the similarity between structures of different organs of different organisms is called homology. Organs of different organisms differ in their functions, but they have the same structural plan and origin. Such organs are called homologous organs, and this term was introduced by Richard Owen. Now, I said we have to do two examples from animals. So first we will do examples from animals. Now in this case we will take the examples of animals like wing of bat, arms of human, paw of cat, and even wings of a bird, and besides this we can take into consideration forelimbs of horse and flippers of seal. All these have similar structure, but the function performed by each of these organs is different. All these organs have similar structure, but function performed by them is different. In case of plants, we take the examples. In case of plants, okay, I said two examples. So one example is this, and second example in case of animals is that heart of fish, frog, lizard, bird and man. All these have similar basic structure, but the function is different because of the difference in the mode of life of each of these animals. So second example is heart of bird, fish, man. All have similar structure but different function due to different mode of life. Then coming to plants, a thorn of bougainvillea and a tendril of cucurbita. Thorn of bougainvillea and tendril of cucurbita. They all have, both of them have similar structure but function performed is different. This is one example. Second example is rhizome of ginger and tuber of potato. They also have similar in structure but they perform different functions. They are both modifications of stem. So this was about homologous organs. Coming to analogous organs. The similarity in functions of the organs of different organisms is called analogy. Organs of some animals have similar functions but they differ in their structure, origin and development. Such organs are called analogous organs. Examples that we take from animals: wings of insect and wings of bird. They are both completely different in their structure, in their internal structure and origin. But in spite of having a different structure, the function performed by them is similar. Both help in flying. So this is one evidence. Second evidence is analogous organs, where we say structure is different but function performed is similar. As we just now discussed, one example in case of animals, that is wing of a bird and wing of an insect. Both have the same function of flying but their structure and origin is different. Second example, fins of fishes and flippers of penguins and dolphins are also completely different in structure and origin, but they perform similar function, that is help in movement of water. Two examples from plants: sweet potato, which is a root modification, and potato, which is a stem modification. They are analogous because the function performed by both of them is storage of food, whereas their origin and structure is different. Sweet potato, as I told you, is a root modification, whereas potato is a underground modification of stem. Second example, tendrils of grapes and tendrils of pea. Again, the function is same. They both help the plant to climb up, but tendrils of grape is stem modification and tendrils of pea are leaf modification. Tendrils, that part of the leaflets that change into tendrils are seen in pea, whereas in grapes we see it is a stem modification. Then next is vestigial organs. Vestigial organs are those organs which were present and had a function in the ancestors, but now these organs are not functional in some organisms. So some structures in the body of organisms are not functional. However, these structures were functional in their ancestors. Such structures or such organs are called vestigial organs. For example, in humans we see the muscles of the external ear, pinna, then appendix, then coccyx, then nictitating membrane and wisdom tooth. All these structures are present, but they do not have any function. So we call them vestigial organs. So the next evidence we will take is embryological evidence. This was analogous and homologous organs as evidence that we had taken for evolution. Next evidence we are going to take is embryological evidence. Evidence is proof. Carl von Baer is known as the father of embryology. He proposed that the embryos of different organisms exhibit remarkable homologous similarities which suggest their common ancestry. Study of embryos of different organisms provides strong clues in favor of evolution. For example, all the vertebrate embryos show the formation of gill slits. These are present even in human embryos. Gill slits are useful in fishes as they live in water, but these gill slits are of no use to land vertebrates. So the question arises, why do these structures appear in the embryos of land vertebrates? And the answer is, if we assume that the land vertebrates have originated from fishes. Baer proposed that organisms which share common descent, that is, those that have evolutionary lineage, exhibit similar embryological patterns on which they later build their adult patterns. This is what is called as Baer's law proposed by Carl von Baer, who is known as father of embryology. Then we come to the recapitulation theory. In embryology, recapitulation theory is important. This theory states is based on the basis of developmental history of animals, and Ernest Haeckel proposed this theory. Ernest Haeckel proposed this theory, and the theory states, this theory is also called biogenetic law. And the theory states that ontogeny, which is embryonic development of an organism, recapitulates phylogeny, which is evolutionary history of the race. Now, ontogeny recapitulates phylogeny, which means that the embryos in their development repeat the evolutionary history of their ancestors. For example, a tadpole of frog is like a fish because it has a tail, it has a tail fin, it has gills, it has lateral lines, which are all characters of a fish. These fish-like features in the development of a frog provide a clear evidence that amphibians have evolved from some fish-like ancestors. We know, we have studied that amphibians were the first terrestrial chordates, and amphibians' name was given because they complete their life cycle in land, in water and on land. So for the metamorphic states to develop, water is essential, and we can see that the larval stages of amphibians, of frog for instance, the tadpole is a larval stage. It resembles a fish in many aspects. So they have an evolutionary lineage. Ontogeny recapitulates phylogeny, which means that the organisms repeat the evolutionary history of their ancestors. Then after, just now, this law states ontogeny recapitulates phylogeny. Which means, as I just now told you, that the embryos in their development repeat the evolutionary history of their ancestors. This is what is meant by recapitulation theory. So now we move on to paleontological evidences. That is, evidences or proofs that we have obtained from fossils. Study of fossils is called paleontology. So the proof or evidences that we have obtained from fossils. Fossils are the preserved remains of plants and animals which were embedded in layers of the earth, and when dug out, we could trace their evolutionary history. So to define fossils, we say fossils are the petrified remains or the impressions of the organisms of the past. Plants or animals which were embedded in a dugout of the superficial deposits in past geological periods is known as fossil, and the study of fossils is called paleontology. Fossils help us to understand the process of evolution by giving clues to interpret the habits and habitats of these organisms. Fossils reflect the history of the past, hence are true witness of evolutions. Fossil of Archaeopteryx, which is a connecting link between the reptiles and the birds, clearly suggests the origin of birds from reptiles. So, one bird, Archaeopteryx, is a fossil bird, and this is a connecting link between reptiles and birds, which clearly proves that birds have evolved from reptiles, because this bird has a body like a lizard and has wings like a bird. So it is a connecting link between a reptile and a bird. Then we come to the next topic, that is geological time scale. Geologic time scale covers the whole span of the Earth's history to correlate the events in a proper sequence. Radioactive dating techniques have revealed that the Earth is approximately 350 million years old, and on the basis of time, the geological history of the Earth has been divided into 5 eras. And these 5 eras are Archaeozoic era, then Proterozoic era, then Paleozoic era, then Mesozoic era, and Cenozoic era. These are the 5 eras in which the Earth has been divided, and each era includes several periods, and each period is further divided into an epoch. Now, in the Cenozoic era, the dominant plant life was herbaceous plants, and the dominant animal life was it was the human beings. It was called the age of man. Then the periods of Cenozoic era are quaternary, tertiary. In the tertiary period, angiosperms were the prominent plants, and mammals were the prominent animals. Then we come to the next era. In the next era, that is the Mesozoic era, the gymnosperms were the main plants, and it was the age of reptiles. In Mesozoic, the period Cretaceous was for gymnosperms, and it was the age of reptiles. Then Jurassic period was dominant for the lower flowering plants, and the toothed birds, reptiles, were dominant like the dinosaurs. Then in the Paleozoic era, the periods that were there were Triassic, Permian, Carboniferous, Devonian, Silurian, Ordovician, and Cambrian. And different forms of plants and animals were seen in each period of Paleozoic era. Then was the Proterozoic era. In Proterozoic era, it was seen that the plant as well as animals were unicellular, and they were simple. I will be putting up a PDF for this geological time scale, because this is more of learning, and the table is also given in your textbooks. So refer to that table. I will be putting up a PDF for this geological time scale. And I think this much is enough for today. We will continue on the next turn. Good luck to all of you.